Well, today we're going to have a special treat, and we're going to hear from David and Ryan, your two GSIs, who both have expertise in biological matters that I don't, and can give you an insight into some aspects of life history better than I can. But I need to tell you first that in each historical trajectory there are trends and there are cycles and there are occasional events. Well, last night there was an event in the history of McCone Hall. You might have noticed the signs on the door coming in. So what happened was that a water main broke and flooded the basement. Fortunately, Bill Dietrich was here, who is a world-renowned professor of hydrogeology and knows all about water flow. And he and some people who also happen to be here, including your GSI, spent half the night bailing and saved the building. So, but what they didn't save was the water to the restrooms. So let me suggest that if anybody has an irresistible urge in the middle of the lecture, that probably the best thing is to go out the front door of the building, turn left, and on the way down the hill on your right is the East Asian Library. And that would be a good place to go.
McLaughlin Hall, too.
What's that?
You can go to McLaughlin Hall, it's really close.
Yeah, McLaughlin is that one. So there are various places around. We're sorry we cannot help you in this building today. Maybe by Thursday. So at any rate, without further ado, here is the first of our lectures for today, David Mangiante.
Excellent.
Thank you.
All right, ladies and gentlemen. Today, Ryan and I are going to be talking to you about the origins and evolution of life on this planet. And since this is a history course, I'm going to sort of take an historical perspective and go through some of the developments and instances in human history that have driven our understanding of life history. But before I do that, I think we should all ask the question, what does it mean to be alive? So here I've depicted a ficus and what I'm going to call a facus. One of these is alive, one of these is most certainly not, but how do we know one is alive? And what does it mean to be alive? And so just to start off, get everyone thinking and excited, I'd like some ideas from all of you. What does it mean to be alive? Go for it, Deborah.
Well, I wondered that very same thing, so I looked for some quick explanations, and one of them I thought was reasonable, which is life is a self-sustaining system of chemicals capable of Darwinian evolution.
All right. What are some others? Other ideas? What does it mean to be alive? Organic system, okay. That raises many other questions, actually. But so what are some other ideas? Yeah.
Maybe some form of growth.
Growth, okay.
Something that responds and reacts to its
Responds, reacts. All right, let's get a couple more. Nobody has any other ideas in the back.
Capable of reproduction.
Capable of reproduction, and one more right here. You were going to say, what were you going to say?
Pretty much the same thing.
All reproduction. Okay, well, that's great. So we can have lots of different definitions for what life is, what does it mean to be alive, but the definition that we can usually find in a textbook on biology really goes like this. It has these different components: homeostasis, organization, metabolism, growth, adaptation, response to stimuli, and reproduction. And all of these have different histories and different developments that have produced them. But we sort of have to ask the question, what is in this definition? And when we look at these words and look at these and start thinking about what is life and how we define it, we have to think about how and why is this definition applied? Where did it come from? And sort of what utility does it have to our lives and our future? And so I'd like to sort of whet your appetite when it comes to thinking about why we like thinking about life. And it comes, and we're going to talk about this later on in the class, but one of the current directions in research in the origin and evolution of life is life on other planets and other objects. And so when we start thinking about what is life, it's sometimes useful to think about when we look at another planet, like this is a picture of Europa, it's a moon around Jupiter, and this is a picture of Titan, moon around Saturn. If we were to go there and look, what might we find? And what might we characterize as life there? But we must also remember that our definition for life is defined really by what's on Earth, the life that we have on Earth. And so I have depicted here the whole grand extent of life on Earth. Walter showed this picture the other day, and I'd like to make a little clarification and explain to you really what you see when you look at this, because some of you may not be familiar, in fact, with this type of diagram. It's called a cladogram, and really attempts to define interactions between organisms, and this particular one is color-coded so that we can break it down more easily. But each of these little lines refers to a certain type of organism that lives on Earth in the current day. And so by looking at the types of organisms, we start defining what life is to us. But for the purposes of our talk here, we're going to ignore most of those textbook definitions, and we're just going to go along with a very simple one, which is that life is reproduction and life is metabolism. You can reproduce and you can metabolize. You are alive. And I'd also like to emphasize this idea that life, typo there, but life follows an arrow and is driven by cycle. That is driven by cycles and punctuated by events. And as we talk through the development of the history of life on Earth, we're going to come through different cycles, different arrows, and different events. So keep this idea in mind that although evolution is an arrow, there are many rich cycles that drive it forward. So with that, we begin the history of the origin of life from the scientific worldview. And so here I should actually mention that when thinking about the origin of life and how life arose on Earth, we should remember that it's actually not exactly a scientific question, because it's very difficult for us to actually find a hypothesis to test. And so there are many different worldviews that can be applied to the idea of the origin of life, and perhaps equally valid or maybe more valid than the scientific worldview is a philosophical or even religious worldview. But in the context of this class, in the context of this presentation, we're going to just talk about the scientific worldview and the advancements that have given us insight into maybe how life developed. So I'm going to start way back. Well, not that far back. We're going to start in the 1800s, late 1800s, with two gentlemen, Helmholtz and Kelvin, two incredible chemists and physicists and scientists in general, gave us many of the theories that we now currently use in the study of Earth. And they were interested in an origin of life that had to do with what might be called the seeded Earth. And that is the Earth that is barren, and then meteorites come and impact the Earth and provide it with organic material and potentially provide it with life. And so the sort of like schematic that I've drawn up is the idea that you can sort of have an event that produces a multitude of life around Earth that then proceeds to slowly evolve. And now Kelvin and Helmholtz understood that evolution was an important process in Earth and life history. And they understood that most likely the original material that showed up on Earth would have had to go through a series of processes that would make it more and more complex. And we're going to carry that idea of increasing complexity throughout the rest of our little exercise here. But we should remember, we should recall a couple things about complexity, and I'll just give you a few little tidbits from complexity theory. So one of the ideas is that to have a complex system that grows out of less complex systems, we need to have an energy source. We need to have something that drives that system in a direction. However, we can't have too much energy. And so if you think about, let's say, a flame and the smoke rising from a flame, initially we have an energy source that produces that smoke, but that energy either dissipates or comes in contact with too many air molecules and breaks apart. And so as we think about complex systems developing, we have to think about how an energy source can drive them, but not an energy source that's too strong to rip them apart. And this will come up later. So Helmholtz and Kelvin have this idea of the seeded Earth. And that's fairly interesting, but we don't really have too much evidence. There isn't too much evidence for that necessarily being the case. And so later on in the 1920s, we get a different style of the origin of life. And it comes from these two gentlemen here, Opain and Haldane. So Opain was in communist Russia. Haldane was in Britain. Both were members of the Communist Party. Both ascribed to the sort of Communist Party idea that life originated on Earth not by sort of like the hand of God, you might say, but instead arose out of a primordial soup. And so these two gentlemen put forward the idea, that initial idea, that life really was a soupy mixture that, through whatever processes, became more and more complex and developed into what we have today. However, this idea of the primordial soup really is kind of difficult to wrap your head around, because if you think about a prebiotic ocean, or any ocean at all, filled with organic materials, we might like to think about how are those organic materials going to come in contact with each other? How is this complexity going to arise? And in the primordial soup, the likelihood of two organic molecules or two molecules or any two species at all coming in contact is very low. And granted, we're talking on the timescales of billions of years, but it seems as though life developed very rapidly on Earth. And so the primordial soup idea is a nice little brain thought, but probably is not a good explanation for the origin of life. But people still thought about it, and thought about how we might improve upon the primordial soup. And so I'd like to take you, and I think Walter mentioned these two gentlemen before, but Stanley Miller and Harold Urey, both of which actually have Berkeley connections. Miller got his undergraduate degree here at Berkeley, and Urey spent his time, and I believe he won his Nobel Prize when he was here at Berkeley. I may be incorrect about that. But both of these gentlemen found their way to the University of Chicago and concocted an experiment where they took the primordial soup and blasted it with energy, lightning in a bottle, put as much energy pretty much as they possibly could into the system to see what they would get. And they found all kinds of interesting tidbits. Many people questioned their experiment, and I, in fact, myself questioned their experiment, since some of their assumptions regarding their prebiotic soup and regarding their experiment itself seemed to not really fit. And if we recall what I was talking about earlier with regards to complexity, if we have a prebiotic soup where there's a lightning strike that hits the soup, what it's going to do is, yes, it's going to produce amino acids, or potentially produce amino acids. Yes, it's going to produce complex organics, but at the same time, it's going to rip them apart. And so we're not going to necessarily be able to arise at this state of complexity that we need to harbor life. And so the Miller-Urey experiment, the Urey-Miller experiment, in some ways has been dismissed as a viable theory for how life is developed. However, I should mention that all of these theories, again, are not really testable and are really thought projects when we start musing about how life could originate. And again, we should harken back to the idea that we want to be able to be thinking about how life might be developing elsewhere in the universe, how life might be developing even under our nose in our solar system. And so ideas like Miller and Urey's are good to keep around, because it's unclear whether or not this sort of thing could potentially lead to life. But all right. So it seems that we have too much energy. And so people started thinking about, how could we have a system that could provide energy to organic molecules and to organic complex systems without blowing it to pieces. And we arrive later. So this development actually happens much later than all of those. So the Miller-Urey experiment happened in the 50s, and thoughts about sort of the iron-sulfur world and later ideas really happened in the 80s and 90s. So these are very recent ideas for how life could have developed. And so I'm going to pause here in the history and sort of talk about a little bit about the character of life, and the character of life as it exists now on Earth. So what I have depicted here is what is called a metabolic cycle. And what I was saying earlier about how life is really, we can really define life as being metabolisms, which means consuming energy, and reproduction. And the iron-sulfur world is a metabolism-based idea for the origin of life. And one way to think about metabolism is kind of like an energy pump, where we take materials, starting materials like water, carbon dioxide, and we drive them through this cyclical reaction, and we pump out the energy into the organism. And this happens over and over and over. And in fact, all or most organisms use, in fact, this exact model for this biological pump, this energy cycle, this energy pump. It's called the citric acid cycle. Some people call it the tricarboxylic acid cycle, and other people call it the Krebs cycle. And it is a way that we as humans use. We use this cycle to metabolize CO2, and we drive it in what's known as the oxidative direction. But we'll get to that all later. But the key here with the iron-sulfur world, the idea here is that we can potentially form an energy sink in a world without having a violent catastrophic event. And we can utilize energy in minerals, so iron and sulfur minerals, in fact, and we can utilize energy on their surfaces to slowly produce this energy pump and slowly build complexity without utterly destroying it. And so this, if you want to imagine what the iron-sulfur world would have looked like, we have to think about a mineral surface sitting in some sort of liquid, and on its surface are all of these organic molecules. And they're interacting with each other slowly over time. And in fact, we can even impose evolution on this sort of cycle. We can say that maybe there are a set of molecules that interact on this mineral surface, and over time they develop and they change, and different molecules come into play, and different molecules in how they interact potentially metabolize energy better than others. And so slowly over time we can potentially have some sort of evolution. However, this idea to think about the iron-sulfur world as like a viable idea for how life might have arisen, we're really missing one of the key components. We can't really replicate it. None of these molecules really would build themselves, and over time, this sort of setup, we have this specific set of molecules interacting in this cycle, would not rapidly reproduce. And so we're left with wanting feeling of we really are missing one of the essential portions of our existence and the existence of all organisms on the earth, and that this thing can't reproduce. And that, yes, it makes a nice metabolism, but we can't really do anything with it. And so, sorry, go ahead. Thanks. All right.
Also, where would the sugar be coming from? Or like the hydrocarbon energy source?
Where would the sugar be coming from? Well, so
Where would the energy source be to run the...
in this case, the energy source is actually CO2 and water, effectively. But in the prebiotic, or thinking about the prebiotic world, we can maybe imagine that it's possible to catalyze reactions that add CO2 together on mineral surfaces. And by adding it together on mineral surfaces, we can build these higher order organics. In addition, there has been some evidence to show that organic molecules can show up simply from reactions in an atmosphere. If you have carbon dioxide, methane in an atmosphere, and there's UV light, you can produce organics. You can produce cyclical organics, so like cyclohexane and things like that. In addition to that, there is evidence that potentially organics can show up on meteorites, and that meteorites falling from out in the solar system can give you some of those sugars and give you some of those building blocks that you would need to break down. But here, most of the energy is actually coming from the CO2 and the water, and in some cases also from the electrons in the minerals themselves. Cool. So all right, we need some idea of how the heck we can get reproduction. Because without reproduction, clearly life as we know it would not exist. And this really doesn't do it for us. It doesn't cut it. Thinking about how DNA would evolve out of this kind of cycle is fairly difficult. And so people started thinking about ways that they could potentially make DNA in this prebiotic world. So we come to the idea of the clay theory in the RNA world. And some of you may have heard of this before. RNA is, many people like to think about RNA as the first life form. I'm going to tell you that that's not really a viable option, but thinking about how RNA might have existed in the prebiotic world is actually fairly fascinating. So here depicted is an RNA molecule. And I've clumped this with this idea of the clay theory because as we think about these origin of life ideas, we have to remember that chemistry is very well catalyzed and produced on mineral surfaces. And so if we can think about how those mineral surfaces interact with organics, we can get some idea for how that complexity that we're striving for could have arisen. There's an interesting character of DNA and RNA. All DNA and all RNA has something which we call handedness. And I'm not going to get into at all really what that means, except that it means that it kind of means that there are two different sets of RNA and two different sets of DNA. One of them you can call as being right-handed, and one of them you can call as being left-handed. And it turns out we only have one of those sets. There are there's only, I think it's only left-handed. I don't remember which handedness it is. But, Ryan, do you recall? Doesn't matter. What matters is that if we add a little bit of the other handed molecule, all reproduction stops. And we cannot function in the presence of the other handed molecules. So the question is, how did this single type of molecule arise? It's still the same. Both handed models are the same molecule. They're just shaped slightly differently. And so that's where the clay theory comes into play. And it's thought that potentially clay minerals, which you're probably familiar with if you've seen talc or even crafted with clay in an art studio. Clay is this sort of silky material that has fine grains. And really what that comes down to is clay minerals are layered. They have these layers. And so right here depicted are two layers of a clay mineral. And in between them there's this space, which is great. There's a space. Okay, we can put things in that space. In fact, we can put bases that build up an RNA in that space and have them self-assemble. And when they do so, they bind to the surface of the mineral in such a way that potentially it produces the handedness that we see in the world today. And so it's hypothesized that actually DNA and RNA were self-assembled molecules on clay surfaces in the prebiotic world. And that's great. RNA is a great, exciting molecule. RNA can catalyze reactions, it can reproduce, and it can do all the things that we really want life to do. The only trouble is, how do we get these bases, and how do we get them into the clay? And that's sort of been something that many scientists push under the rug. But again, remember, we're thinking about potential ideas, and these are not hypotheses that can necessarily be tested, but instead are directions to think about how life might be developing. So, our two sort of current theories for how life could have developed are the iron-sulfur world, metabolism-based, metabolism-centric cycle, and the RNA world, which is a reproduction-centric cycle. So to compare the two of them, the iron-sulfur world idea is what's known as autotrophic, which means that it can live on its own. It can produce its own energy. It doesn't need to consume molecules from the outside environment necessarily. It's a reliable energy pump. It can cycle through and pump energy through. You can sort of think about it kind of like as Walter mentioned, the Sahara pump, with the wetting and the drying, pumping people out of Africa. This, in many ways, is sort of pumping energy through by cycling around the chemistry. And also another interesting aspect is that in many modern metabolisms, there are metal centers. There's all this metal in many of the reactions that happen in our bodies now. And thinking about where that metal might have come from, it could have come from this sort of iron-sulfur world. But as I mentioned, it really has no reliable means of reproduction. So it's not that viable of a life form. So the RNA world, though. RNA, information rich. This RNA and its pal DNA are in many ways the lifeblood of what we are. And so it's full of all that information. It's reliable reproduction, and it's ubiquitous in modern organisms. However, like I said, it lacks a viable energy pump, and also it is effectively heterotrophic, since to have an RNA system, you have to keep consuming bases, and they need to be coming from somewhere. And in the prebiotic soup, as I mentioned, two molecules interacting is fairly unlikely and difficult. So both of these have their drawbacks and seem as though maybe neither of them were how life might have developed. But of course, we can reconcile this. And we can think about why not put them both together? Why not have a metabolic cycle functioning on a mineral surface, coming in contact with RNA that's been produced in a clay mineral, and through a variety of other interesting, exciting means, which I'm not going to get into right now, they come together inside of a cell. And so now we have maybe a metabolic cycle plus a reproductive cycle put together, making what we are really defining as life. Yeah.
Now, what do you mean exactly by cell? Because I don't think you mean cell wall.
Right. So something actually interesting about cell walls. Cell walls themselves are self-assembled, as in if you get cell membranes. I'm sorry. Yeah, not cell walls at all. Cell walls are way later. Cell membranes are self-assembled. They have these big, long hydrophobic bits, so they like to stick together. And so perhaps what may have happened was that in this origin of life scenario, we have these little bubbles of cell walls floating around, which could have potentially encapsulated some of these things. But yes, I'm talking about mineral surfaces. I'm talking about a collection of minerals in proximity that these processes are happening on. How you make the leap between that and an actual cell, if you can find that out, that would be awesome. But yeah. But now we have something that's reasonably viable, and that's exciting. That means that maybe we can start thinking about environments that facilitate this clay hypothesis and think about and facilitate this iron sulfur hypothesis as we look at other planets and other objects. Of course, there are many other ways that we could potentially have life, as I already mentioned, and so we need to keep our eyes open and keep thinking. But I'd like to come full circle, and so I went through. Oh dear, I went through sort of the evolution of these two ideas, the iron sulfur and the RNA. But I mentioned that we started with meteorites, and I mentioned that organic material can come from meteorites. And just recently, in fact, a few days ago, there was a paper put out that sort of summed up a bunch of data from meteorites. And so what I have depicted here is a carbonaceous chondrite, which is a specific type of meteorite, and some of the fun things that have been found in it. So this right here is an SEM image, a scan. It's a zoomed-in image. This is the scale bar. It's 1 micron, very tiny image of this shape. They looked at what the elements are. They looked at what might be going on, the morphology, and they compare it to this shape. They look fairly similar. One of these shapes is a meteorite. One of these shapes is a bacteria. And they think that potentially this meteorite was host, or at some time in its evolution was host to life, to microbes. Now, that's very cool and very awesome, and it'd be thrilling if we found life on other celestial objects. But it's not always entirely compelling. And it's important to remember that these sort of arguments are not necessarily proof that this is how life arose on Earth, and are not necessarily even proof that this, in fact, ever was life. Since my advisor in particular will give you, a laundry list of explanations for how this object is entirely different from this object. And depending on your philosophy and depending on what you want to believe, you can decide whatever you like. However, this is another idea for how life might have showed up on this planet. All right, so we've been talking about the direction of the history of producing, or of the origin of life, and we've been talking about some cycles that are apparent in life. And if we recall, I mentioned that life is an arrow driven by cycles, metabolic, reproductive, but it is punctuated by events. And those events are often where a lot of the interesting things show up. So I'd like to talk to you very briefly about 2 exciting events in life history and Earth history. The first of them is the rise of oxygen. So some of you may know that oxygen in the atmosphere, oxygen was not always present in the atmosphere. And most of the early Earth was what's known as a reducing atmosphere, devoid of oxygen. And so all of this part of history, life is developing, figuring out how to reproduce, how to metabolize, finding niches, doing all of these exciting biologic things without oxygen being present in the atmosphere. And then suddenly, one little guy figures out that it's possible to harness light from the sun and produce oxygen. So this is a little schematic diagram of a cyanobacteria, and here are a bunch of them. So cyanobacteria produce oxygen. And it seems as though oxygen arose fairly rapidly on Earth. And there's a good amount of geologic evidence for that. Some of it is shown here. That I chose this one. This isn't actually maybe the best explanation for oxygenation, but it's maybe the prettiest. This is called a banded iron formation, and the red here is hematite, which is oxidized iron. And so in the ocean, non-oxidized iron floats around. But when it gets oxidized, it forms a mineral and sinks and beds like this. And so we find these banded iron formations, or BIFs, and we can date them in interesting places, but they represent, or we think they represent, oxygenation events in the oceans of the early Earth. But this oxygenation, so this event, okay, it's kind of interesting. It's interesting that we now have a different gas hanging out. But what does it mean? If we recall the way I didn't really mention how these metabolic cycles, this metabolic cycle works, but this metabolic cycle only works in this state without oxygen. With oxygen, it gets ripped apart. Oxygen is very powerful. And you may know that if you were to breathe just straight oxygen, you would die. It's very flammable. It's very reactive. And there's a reason that we eat antioxidants. And so all of that life that is developed, all of this 1.5 billion years or something like that of life that is developed is now in the presence of oxygen, and is now potentially getting ripped to pieces. And maybe this is the single most devastating mass extinction in the history of life on this planet. And who knows, if oxygen hadn't arisen, perhaps we would have had a whole different history after here, since life effectively, in many ways, had to restart itself. So the last event that I wanted to tell you about was this event called endosymbiosis. And Walter mentioned this the other day, but I want to talk about it again because it's so epically cool. Endosymbiosis is the idea that one organism can consume another organism, but instead of breaking it down into its parts, they in some way live in harmony with each other, one producing energy for the other or doing a function for the other. And so it's thought that most, if not all, of the organelles, or the pieces of the modern eukaryotic cell, are products of endosymbiosis. And this added tier of complexity may have been what springboards us from this very the slow evolution here through Eubacteria archaea into our modern crazy diversification of life. And I know Ryan will have many more things to say about that and how that's happened. But endosymbiosis represents a new tier in the development of life. And we move simply beyond just adding complexity internally to actually going out and finding and getting complexity. So with that, I'd like to end and turn it over to Ryan.
So, am I out of the way here? Yes, I am. So we promised you a lecture about the history of life on the planet, and so I feel like David had the hard part. David had the part that doesn't involve pictures of plants or animals or what you usually think of as the kind of like beautiful pictures of life on the planet. And also this is not David's fault. It's, I think, inherent in looking at very early life on the planet, where there are just tons of kind of hand-wavy explanations for things and tons of just unsatisfying ideas. And in part, that's what makes it fascinating, because it's just such an insane leap to think about how to go from non-life, how to go from a handful of chemicals in a soup or on a mineral surface, and how you then end up with families of chimpanzees living in trees today. That is an amazing series of leaps of complexity through time, and it's really hard to wrap your mind around that. What hopefully I can convince you of here is something that's a bit easier to wrap your mind around. It's once we get started with life, once we have cells, then how do we make the next kinds of leaps in history to build complexity towards what we see today? So this is a very ambitious agenda for the next 35 or so minutes, but a brief history of multicellular life on the planet, evolution, and a little bit of molecular ecology. So I don't have a pointer really, but that's okay. This is our slide. This is our agenda here. So how do we think about building? David did a great introduction, building complexity within a cell. So that's the endosymbiotic event in history. So you're going from prokaryotes to eukaryotes, things that have no nucleus, to cells that have not only are bounded by a membrane of some kind, but have a nucleus on the inside and have a mitochondrion in the inside, both of which seem to have been free-living bacteria at some point in the past. So that's the evolution of complexity up to about 2.5 billion years ago, or thereabouts, 2 billion years ago. And so what I'd like to do is kind of take us to, so how do we build from that cell into multicellular organisms? And then once we have multicellular organisms, how do those multicellular organisms become more complex themselves, rather become more diverse themselves, diversification of species through time. And then a little bit how we know, a little bit about how we know what we know about the history of life on the planet, and then how we're looking at that right now, because of course diversification isn't something that just happened in the past. Diversification is something that is going on constantly. So here is, you guys may recognize this, I believe I pulled this from Chronozoom last year. This is the kind of time history of life, and David used the same figure, but what's missing here are some of the dates because it got cut off at the top. So this is the last universal common ancestor at 4.2 billion years ago, it looks like. And then the eubacteria, you get the endosymbiotic event at somewhere in the neighborhood. Actually, I don't have a date for that. This looks like 2.5. So let's call this somewhere in the neighborhood of 2 billion years ago for the endosymbiotic event. So what I'm going to be concentrating is all here. So this is all going to be smashed up here. But notice how the tree of life starts to diversify a lot once you get into symbiosis. And that's probably not an accident. You probably build up a lot of diversity after that point. But the other thing you want to think about is we know a lot more about the recent than we do about the deep past. So there are no fossils at all, really. I mean, that's not exactly true. So Jerry Lips will tell you that there are fossils. But what he's going to talk about as fossils in a few weeks are fossils of a cell or a couple of cells or mats, bacterial mats and things like this. You don't get hard parts until about here. So there's this huge history of the planet where we don't have tons of physical evidence. We've got to reconstruct what we know from circumstantial evidence. So multicellularity is this, you get this really interesting philosophical question of what's an individual. So if you have one cell and you've got your cell and you're hanging out here in this ocean, and there are some other cells around, it seems pretty clear what an individual is, right? An individual is the one cell, and it's different from each other such cell, and that is an individual. But if you start to get many cells hanging together, and you get to be a multicellular thing, it's less clear what an individual is, right? Because there are some individual characteristics of the cell and individual characteristics of the group of cells. And so that's going to be a theme that we'll come back to. So one take-home message here is multicellularity arises a whole bunch of times. And so far, we know of about 25 times that multicellularity has arisen in the other branches of life, not animals. So look at the mostly, I think about plants and fungi in this regard. There are multiple times where multicellularity has arisen independently in those lineages. It seems like it's fairly easy to do over evolutionary time. But for animals, the group that eventually leads to animals, of course, of which we are a part, has only one origin, one multicellular origin. So this is really interesting, I think, and I've been trying to learn more about this because it has really important effects for development, important implications for development, how animals develop, what constraints there are on evolution. So I'm going to get into that right now. If multicellularity is easy to do, it's interesting that it evolves only once in the stem, in the lineage that ends in animals, or that includes animals. And the idea behind this is that our common ancestor, this is going to require you guys to remember a little bit back to high school history and mitosis, which may be kind of a bad word in some of your minds if you're a humanities student and you've been trying to escape mitosis in college. We're going to drag you back in very briefly. So in mitosis, as you might remember, cells divide, and that's the process, one of the processes by which cells divide. And in that process, you get here, this is a cell undergoing mitosis, and so the red is the chromosomes as they're being pulled apart. And the structures that pull apart the chromosomes are dyed green in this picture, and that's called the spindle, the mitotic spindle. And these are proteins that, my understanding is that they're kind of proteins that link together. They're very strong, kind of fibrous things, and they're like the utility infielders of the cell. They can do a bunch of different functions, but here they're functioning to kind of pull apart the chromosomes during mitosis. So it turns out that these structures, the spindle, are homologous with the cilia and flagella, which are the ways in which cells move about. So in case you are unfamiliar with any of the words that were in that last sentence, here is how that works. If you've got a cell, and some cells you may have seen have kind of a bunch of little hairs around the outside, those are called cilia, and those all beat simultaneously or in some pattern, and then the cell can move along that way. And alternatively, sometimes cells have a big tail, and that's a flagellum, and they might have multiple tails. But that's another way of locomotion, another way that cells will move through a fluid environment. So the really interesting thing is, as I said, the mitotic spindle is homologous to the flagella and the cilia. That is, these are actually the same structures, evolutionarily speaking. They have a common origin. So in the group that leads to animals, in our common ancestor, the common ancestor of all of these people in this room, and our common ancestor with all other living animals, our ancestral cell that led ultimately to us could either use that material as spindle, or it could use it for mobility. It could use it as a flagellum or a cilia, but it couldn't do both simultaneously. And the reason for that is that there's a mechanism in the cell where it couldn't, effectively it kind of couldn't make both simultaneously. It would recycle them. And this isn't just in history, this is actually now. You can look at animal cells, and they can't do that. There is no example of an animal cell that is ciliated that also divides. Is there a question up front? No? Okay. So it leads to this really interesting conundrum, right? Because if you're a cell and you're hanging out in the water, and you are trying to live your life, you're doing your thing, it would seem really important that you could both move and be able to eat, because cilia are going to be involved in getting to food, finding food, engulfing food. you need to be able to do that, but you also need to be able to reproduce, right? Because as David was just saying, part of the what we think of as being alive is a requirement for reproduction. And reproduction, if you're a cell, means division. That's the only way to do it. So here you have this kind of grand conundrum. If you only got so much hardware, and you can either move and eat with it, or you can reproduce, but you can't do both simultaneously. And so I was trying to think of a good metaphor for this. This is kind of beyond our experience as humans. We don't run into things like this very often. And so my metaphor was this. Maybe this isn't very good, but I'm trying it out on you guys for the first time. So picture if you have a fork. You've got a fork, and you have to but you don't have any legs, and you don't have a lot of appendages, and you're just trying to kind of inch along with your fork to move to where you might find food, and then use that same fork to feed yourself. Or you could use the fork for reproduction. And my example of this, actually, I'm glad that John is here, because this involves his daughter, as a matter of fact. John's daughter in college went out and bought a Mickey Mouse waffle iron. And her idea was that this would help her attract mates, because she could always say, hey, do you want to come back to my place because I've got a Mickey Mouse waffle iron and we can make waffles? And who's going to say no to that? So this is the example of if you've got a fork and you're kind of trying to use that as a way of attracting mates. In Kate's case, sadly, it didn't work, but but it was a brilliant idea, and I'm glad for the concept. So that's the trade-off. You can either eat and move, or you can divide and reproduce, but you can't do both. And so multicellularity is kind of a brilliant solution to this problem, right? Because instead of being able to do one or the other, you can do both. How do you think that works? That's a very broad question. I should have posed that differently. I will tell you how it works, then I'll ask you another follow-up question. How about this? So if you've got one cell that you've designated for movement, you could have another cell that's designated for reproduction, and they could hang out together, and now you have an individual. Let's say you could have a couple of cells, and you could have some of them move and some of them reproduce, and then we could call that whole thing an individual. And then here you have, this is kind of the beginning of multicellularity, right? You've got your bases covered. You can eat, you can move, you can reproduce. And this is something we could recognize as an organism, a simple one, but an organism nonetheless. So you get specialization. But in a fluid environment, what you'd want to do, inevitably, surely there's a tool for erasing chalk off of the board. So let's say you've got this kind of bunch of cells that all band together. And everything is doing fine. They can all be ciliated, and they could kind of move around. And you want to designate some of them for reproduction. Which ones would you choose? Like, where would you put those reproductive cells? And this is not even a rhetorical question. This is a real question. If you were you, and you were making this decision, where would you put the cells to reproduce? In the middle. Why? Because it's protected. Any other ideas?
If it was outside, then it could bud, or at least attach to a mate.
If it was outside, I'm repeating this for the viewers at home because I have a microphone. If it was outside, it could bud or encounter a mate. Josh?
If it's inside, then it can expand whatever the organism or whatever it is, so it can grow easier.
If it's inside, it could grow easier, it could expand. So one thing I find kind of comical, this is mostly for comedy value, I think, is let's say you put your reproducing cells on the outside, as Wes was saying, and then those reproduce, but remember you can't reproduce and be ciliated at the same time. So you do that, and we're going to take away the cilia from a couple of these, and then you've got like half of your ball or most of your ball ciliated, but some of them not. This organism can really only go in this direction, because all of its engine is around the outside, but none here. It actually hinders movement if that happens. It binds you. And so sure enough, it seems like putting them on the inside would be a good idea. And historically, that seems to have been the case, because this is what we end up with. We end up with a hollow ball of cells, which you may recognize as your own baby picture, very early in development. A hollow ball of cells. I believe this is yes, it is blastula. And then you want to put some dividing cells on the inside. The way you do that if you're a ball of cells is you kind of make an indent like this, and this is what developmental biologists call gastrulation. This is the kind of first developmental stage that you're going to see. And you're not just going to see it in humans or chimpanzees. You're going to see this literally in every animal there is, because we all share this common ancestor that had these developmental constraints. So I think this is totally fascinating. So when you create this new layer of cells by bending inwards like this, and creating a gastrula, this is an early gastrula and a late gastrula, you create different layers of cells that then can differentiate. And so we get an ectoderm and an endoderm, which that is a two-layered organism. And cnidarians are two-layered organisms even today. Cnidarians are jellyfish, anemones, things like this. Those have two cell layers, and this is why, because they have this developmental stage, and then they kind of go into jellyfishdom from here. We and other animals, kind of on the evolutionary tree, diverged from more derived, since cnidarians, have three layers, because the mesoderm, the kind of in-between layer of cells, it differentiates as well. And so this might seem like a trivial, you might not think that gastrulation deserves an exclamation mark ever, or in this slide at the very least. But this is super important, because you now have all of the tools that we kind of take for granted. You can do all of these evolutionary important things simultaneously: eat, move, reproduce. And this is the kind of grist for a whole bunch of evolution to act on different specialized germ cell layers, if that makes sense. Walter?
Right. Something that's mystifying me is if the precursory cells that went to become the multicellular organism, if they could either divide or move and eat, but not both, how did they survive before they got into multicellular arrangement?
So the question, I'm going to repeat this for the videotape. The question is, how did cells survive before multicellularity if they could either move and eat or divide? And I think it's an excellent question, and the only answer, I'll start by saying I have no idea, but I think the only answer I could come up with was that there's a lot less competition when nobody can do that. And also, I should point out that it wasn't that nobody couldn't do that. There's at least one too many negatives in that sense. But remember, multicellularity is arising many times, and in a lot of those lineages, they can simultaneously eat, move, and reproduce. Plant cells can do this. Fungi, basidiomycetes, the slime molds, can do this. So you would think that there would be a big selective advantage for those lineages that could do all of those functions simultaneously. And I think maybe the answer is we're lucky to be alive, in the sense that it's kind of amazing that the lineage that had this evolutionary constraint wasn't kind of competed out of existence a billion years ago. Yeah. So to put a date on this, and this is if you're looking at Chronozoom, this makes it easy to kind of see where the dates are here. But two germ layers, the split with Cnidaria and the rest of the metazoans is at 1.2 billion years ago, and three germ cell layers at 1 billion years ago. So basically, this differentiation of multicellular animals is within the last billion years. And that's less than a quarter of the life of the planet. So this is really fairly recent. Now, another jump here in complexity is, remember I mentioned this evolutionary dilemma of if you have, here we now have our developing organism that's made up of a bunch of individual cells, and then if we're going to call the individual the group of cells, a multicellular organism, you get this evolutionary conundrum in that there's a competition among cells within the same organism. Right? Because if evolutionary fitness is passing your genes on to the next generation with as much frequency as possible, then each cell, the selective advantage for each cell would be to divide as rapidly as possible and leave as many daughter cells as possible. But that would kind of mess up the individual thing that we have going on here, right? The collection, the individual organism as a collection of individual cells. That was too many uses of the word individual, I apologize. But you see where the tension is, right? What's good for the cell isn't necessarily good for the overall organism. And this turns out to, I think, and this is kind of the leading thought here, is that our very early development, and the very early development of all animals, gastrulation, and then the division into cell layers, and then the development past that point, is a compromise that's been worked out so that it's simultaneously selectively advantageous for the cell lineages, each of the individual cell lineages, and for the organism as a whole. So this kind of complex dance of very early development seems to be this compromise that got worked out long since, 1 billion years ago, and then hardwired such that we have constraints. We can't just develop willy-nilly from a single cell to an 8-cell stage to a 16-cell stage and so on. There is a series of steps that we take, and those are very circumscribed. And it seems to be that this is why there are only so many solutions that will be simultaneously good for the individual cell lineages and the collection as a whole. Louis. Excellent. Yeah, so Louis' question is, if all the cells have the same DNA, who cares who reproduces, right? Because what's good for one is what's good for the other. And it turns out that they do all have the same DNA, but there is, without getting too far into it, every time a cell divides, mistakes will be made. So that you will have, even within your body, you have a genetic, you have an identical complement of DNA in every cell of your body, but not exactly identical, because some mistakes were made along the way in dividing to make more skin cells, or dividing to make more liver cells, or whatever. And so there are mutations that are called somatic mutations that arise over time in your body that mean that each cell isn't exactly the same. And so you're absolutely right in that there's not a ton of competition between cells because essentially they have all the same complement of DNA. But there are some differences that arise. And so there would be some evolutionary pressure to kind of pass on those changes as opposed to changes that may have arisen in a different cell lineage. So this is developmental conservation is the result. Oh yeah, sorry.
Can you also see developmental conservation in plants? I mean, did it develop kind of independently of the way it developed in animals, or how did that work?
Yeah, that's a great question. And my understanding is that, yeah, early plant development is also very kind of, it's almost like kabuki. There are a set number of moves, and you have to do them in exactly the right order, or else you don't survive. And my understanding is that plant development is like that, but a little more plastic. But I don't know enough about it to say more than that. Josh.
I was just getting this thought that these are kind of like the British Empire with the little, during the Middle Ages with the little kingdoms within it. Each of those kingdoms has the soldiers, the knights, and then they have their families inside, and then they fight for position within that particular kingdom, and yet there's this whole entity that they are to share together.
That's a perfect example. And so the question of, it's really any collective action problem is going to be about the same. What's good for the individual units and what's good for the kind of larger collection as a whole. This is a famous drawing by Ernst Haeckel, who was kind of the founder of developmental biology. And these are all vertebrates, but I'm using this slide to demonstrate how similar development is within the vertebrates. And this is late development, so it's cheating a little bit, but it gets the point across in that this is a fish, this is a chicken, and this is a pig, and here's a human. This is early, middle, late development of these different vertebrate species. And so if you look at early development, we look exactly the same. I mean, they're almost indistinguishable among all vertebrates. So this is just to underscore that we kind of have a common programmed early development that then diversifies later on in ontogeny, in development. So that is so this leaves us with less than 15 minutes to get through the last, half a billion years of evolution. So I'm going to do my very best. And we're jumping from looking at building complexity by joining cells together from single-celled to multicellular complexity, which is a big jump. And now we're going to look at diversification of species through time. So now we have a bunch of different species of multicellular animals and plants as well, and fungi. But I'm going to focus on the animals because we know the most about them from the fossil record. And how does the diversity there change through time? And there are just a couple of slides here, but an amazing story nonetheless. So you've heard a couple of references so far in the class to the Cambrian explosion, and now we finally get to it. This is essentially the most exciting series of events in the history of multicellular life on the planet. Maybe I'm biased, but that's what I think. So on this timescale, and now we're running into the linear timescale problem that Walter frequently refers to. So this is the last 540 million years or so, and everything we're going to talk about now is going to happen in this chunk. So this isn't the best map to use, best chronology to use probably to look at it, but I just want to orient you. So this is where we are in this slide. So the Cambrian explosion, 540 million years ago. What happens is if we get multicellularity starting at 1.2 to 1 billion years ago with the different cell layers, complexity starts to arise, but very little happens in the fossil record for another 600 million years, which is a very long time. So many generations of all of these things that aren't leaving a lot of fossils. So, and then all of a sudden, life on the planet just goes completely insane as far as anybody can tell. And right about 540 million years ago, you get this spike in diversity where all of the major forms of life that exist now in the present day arose by the Cambrian, by this point in time. And there's a very famous fossil assemblage up in British Columbia called the Burgess Shale. It's not the only one, and in fact I'm not even sure it's the best one any longer. But for many years, most famous fossil example of this, where all of a sudden it's a soft sediment, kind of mud bottom, oceanic, shallow sea environment where that was rapidly buried and so preserved everything very nicely. And you have this incredible diversity of life that shows up right away. And so as I said, all of the major types of life that we have now showed up then. So that is, we had cnidarians, we had echinoderms like sea urchins and sea cucumbers, we had arthropods like that now we know as insects and crustaceans and whatnot. We had all of the diversity of animal life, all of the phyla occur in the Cambrian. But also, a whole bunch of other things occur in the Cambrian that haven't made it now to the present day. These are extinct lineages that we have no idea what to do with. Excuse me, we have no idea how to classify them. And in fact, biologists call these problematica because we have no idea. There's a genus here. One of these, this thing. This is the fossil. We don't know if this is right side up. There are a couple of different reconstructions of what such a thing might have been, but the genus of this thing is Hallucigenia. As in, how crazy is this thing? This looks exactly to me like one of the flying toasters that used to be on the Macintosh screensaver. This is genus Wywaxia. There's just this insane diversity of things. God knows what this thing is. It looks like maybe a mollusk of some kind, but who knows? So these are all fairly small. This is the scale bar, 10 centimeters, to kind of show you what that is. And actually, I think David used this an illustration of this same assemblage in one of his slides. So this explosion of things that diversifies in the Cambrian, and so this is. A pervasive mystery in biology is, why did this happen? Why did we have essentially stasis for 600 million years, or if not stasis, at least nothing well recorded, and then everything diversifies all at once? Why would that be? So this, by the way, is what's known as the Phanerozoic diversity curve. Very famous figure. This is from Raup and Sapkowski's work, two guys at the University of Chicago who assembled this massive, massive database of fossil data and kind of allowed everybody to ask these big time-scale questions that we can do now. Along the x-axis here is time, with older on the left and now on the right, and the letters are for the different eras. So this is Cambrian, Ordovician, Silurian, Devonian, etc. So as you see, this is the Cambrian explosion, which is really impressive, starting from very little, but actually not that impressive in the history of diversity of life on the planet. Number of genera, by the way, genera is the plural of genus. So we've seen that a couple of times, but in case that wasn't clear. So couple of and by the way, I fit this Phanerozoic diversity curve is this period of time right here, the period of time that's multicolored on the map, and that's because we've got a bunch of different names for a bunch of different periods for a bunch of different fossil assemblages. So why did this happen all at once? A couple of different ideas. One was already mentioned in class a couple weeks ago, maybe, known as the Red Queen hypothesis. That is, from Through the Looking Glass, Alice in Wonderland. From Through the Looking Glass, the Red Queen, there's a scene in which she's running, and not getting anywhere. And that's the idea, is that you are running, you've got to do an awful lot of work to stay where you are, relatively speaking, with the other species. And so the development of hard parts is an example of this, where if you get hard parts, another species ends up evolving tools to break your hard parts. So then you get harder parts and maybe bigger tools to break their tools. And it's this kind of evolutionary arms race. It's also thought that eyes played a big role in this, that maybe once you could see things, visual predators arose. And once you could see things, you can kind of go after them. And so you had to develop defensive measures, niches for hiding in cracks or becoming camouflaged, etc. And there would have been selective pressure to diversify in that way. Another interesting idea. My personal favorite idea is that you get the development of developmental regulatory genes at this point in time. And so we were just talking about, if you've got essentially an identical complement of DNA in every cell of your body, how does one cell know that it's an eye cell and another cell know that it's a toe cell or a liver cell or a pancreatic cell, whatever? Well, if you've got, and I think probably you guys know this because this is kind of commonly taught now, but this wasn't clear as of kind of 15 years ago. But you have a series of switches, and the difference between an eye cell and a liver cell is not which genes are there, it's which genes are turned on there and when they're turned on in development. And those switches are controlled by a very small handful of genes called developmental regulatory genes. The most famous of which are called the Hox genes, H-O-X. And you can look that up and find probably thousands of papers at this point written about Hox genes. But here's a great example from a paper in Science about 10 years ago. These are the different arthropod lineages. I put some pictures here. This is the Chelicerata, which includes the spiders and scorpions, crustacea, and insecta. Here I've got a wasp for the insects. And you've noticed that arthropods are these kind of segmented body types, right? Segmented body types, they may have appendages on each segment. You can have fused segments sometimes. Sometimes you have multiple appendages per segment, but they're kind of variations on a theme. And it turns out that the variations on that theme are driven by turning these Hox genes off and on. And you can, and that's what these colors are here, which Hox genes are turned on to control fore to aft patterning in the arthropods. So if you would like a wasp that has 15 segments and a set of legs on each segment and maybe some wings on a couple of segments, we can do that at this point in time. It turns out that a wasp isn't really different from a spider, from a crustacean. They're not really different that much, other than turning off and on these regulatory genes and expressing legs in one place or claws in another place, or extending the number of segments. So completely fascinating work that is just a handful of genes leads to all of this diversity. And those genes appear to have arisen in the Cambrian. So to me, that's a really compelling hypothesis for how that diversity arises. Another burning question in my mind is, why haven't we seen any new phyla since the Cambrian? We've had 540 million years. Are there only so many ways to be an animal, and were all of those ways tried out by 540 million years ago? It seems unlikely, but we have no new phyla arising since the Cambrian. So I think that's a really interesting open question. Deborah?
Who turns on genes that turn on the genes?
Who turns on the genes? Who is watching the watchers? That's a great question. And I have no idea, is the answer. You have probably triggers from external cues that turn on the regulatory genes, and that may have to do with methylation, which is one way in which genes get turned on in the cell. You get external cues that result in physical changes that then result in a cascade of things being turned on. So, best answer I can give you. So I have 1 minute left here, and so what I'm going to do is skip. This is, want to put a plug in for one of the coolest websites. It's all these people with science tattoos. These are the truly sick. I'm going to leave aside for right now how we know what we know about evolutionary history, and want to very quickly remind you that this is still going on, that diversity doesn't just stop in the fossil record, that as we speak, evolution is working on all living organisms simultaneously. And when we think about how new species arise, we now have these great tools, to go in and look and say, are these two populations diverging in such a way that we think they might become new species? This is an example. This is a mollusk called a chitin that I worked on. And what we thought was one species turned out to be three. One up in the Pacific Northwest here, and these two split by the Baja Peninsula. This is known as allopatric speciation. That is geographic speciation. Really easy way to think about how diversity arises in the here and now is that things live in different places and therefore don't reproduce with one another. Last one I want to put this is your tie to UC Berkeley. Actually, third tie to UC Berkeley today. Lynn Margulis is the woman who came up with the endosymbiosis hypothesis. Oh, and the Millionieri. So Margulis also is the woman who came up with the idea that there's this evolutionary trade-off between in the animal ancestral cell between eating, moving, and reproduction. So this woman, just a phenomenon. She's at the University of Massachusetts now, but she did her PhD here at Berkeley. This is Hopi Hoekstra, much more recent, a Berkeley undergrad who's now at Harvard. And she was a Tridelt, I believe, as an undergrad at Berkeley, IB major. And she does some fascinating work linking selection on mice in the wild to their coat color, dark and light coats, etc. But more than that, she's found the individual genes that control coat color in the mice. And more than that, she's found the individual nucleotides in the individual genes that are subject to selection, that if you can change one way or another, will change dark mice to light mice, and seem to have been animating the divergence of all of these different, what are now subspecies or species of mice along the Gulf Coast due to single or very few nucleotide changes in their DNA. So really a person who has nailed linking pattern to process in evolution. So I'll stop there. Thank you guys so much.